and she does a lot of work looking at more the molecular scale. And next, then we'll look at the cellular, and then maybe a tissue level scale. So,
All right. Thanks for having me. So, can you hear me from the back? Okay. Hopefully, I can keep on speaking this way. Okay. So, welcome to Illinois if you're visiting. So yeah, like Dr. Bopart introduced, I'll be talking to you about molecular imaging, which is probably the smallest scale of imaging that you will encounter. Okay, so yeah, don't pay attention to the topic. This is the biological pathway that we try to investigate using the single molecule technique. So I'll introduce about it later on in the talk. So first I'll go over the techniques, how the assays run, why we do it, and how we achieve the molecular imaging. So that'll be the first, like, maybe third or third of the talk, and then I'll go into more biological applications of how we then use this tool to study biological pathway, which is the RNA interference pathway. Okay, so here's the scale map. So it's probably too small of a font. So we start with atoms at 10th of nanometer. Then we go into nanometer scales of nucleotides and nucleic acid, and then of course in nanometers and microns we have some organelles and some cells, and then we go into millimeters and centimeters where you're talking about tissue. So the scale that we are at is right here in the pretty much single-digit nanometer scale. So yeah, this is a dimension of molecules that we look at, tens of nucleotide or base pairs of DNA, for example, or individual proteins which can span, I don't know, 50 to 70 angstroms or 5 to 7 nanometers. So that's the kind of the length scale that of imaging that we do. So these include individual molecules, sometimes molecule-to-molecule interactions, or sometimes molecules inside of a cell. But we try to focus on visualizing individual molecules to get more quantitative idea about various interactions and pathways. All right. So in this morning's talk, I guess you guys will see span of this length scale in different talks. All right. So I always start my talk with this slide. Why bother to look at individual single molecules? So I'm a musician, so I'm sure many of you play instruments. So it's, I like using this example. So in my view, like biological pathways or biology as a whole is like an orchestra. There are a lot of different players that play different instruments of different sounds, pitch and timbre, and different rhythm and harmony, but yet they all play beautifully together in harmony. While we can listen to the entire harmony and learn about the flow of the music, sometimes it's helpful to zoom into a single player and see what melody he's playing. Sometimes two in conjunction, right? Then we can single out their sound. I don't think this will work here. Let's see. You do have a clip. Yes? Does the sound work here? No? It's okay if it's not then. It's just there's a little music clip, but I will need it later on. So by doing so, then you get to learn about how he and she together, the interplay between the two players, as well as how they contribute to the entire music. So that sort of justifies why we try to look at single molecules. By zooming into single molecules and molecular actions, we get to learn about their individual role and how they contribute to the biological pathway. Okay, so because they're so small, a single-digit nanometer scale, we have to visualize them somehow, and we like using fluorescence. So we basically tag different molecules with fluorescence of different colors. Sometimes I'll showcase each of the case throughout my talk. Sometimes one color of fluorophore is enough to look at molecular interactions. Most popular technique in single molecule by far is FRET, or forced resonance energy transfer, which I'll also go over in a few slides. And FRET can be expanded to 3 colors or 4 colors, depending on the fluorescent dyes that you choose and how you arrange the dyes along the molecules. In terms of arranging the molecules, a lot of times we immobilize substrates such as DNA or RNA onto surface and add protein or any interacting molecules to study the interaction between the two. Vice versa, we can also immobilize protein and then add substrate to study interactions between the two. And there's a new technique, it's not new anymore, but it's a newly developed technique called single molecule pull down, where this allows you to pull down molecules of your interest directly from cell lysate. So this is really handy because you pull down molecules directly from cell lysate, the molecule-to-molecule interactions, such as cofactor binding or oligomerization of the proteins are preserved. So this is very useful, and yeah, I'm still amazed that this actually works, but it works really well. So I'll show you again each of these cases throughout my talk. So you can interrupt me at any time point. All right, so this is the one-color assay I told you about. I'll introduce the name a little bit later because this is a description of what we actually observe. So imagine a DNA with a fluorescent dye attached at one end. So this is supposed to depict double-stranded DNA, should be helical, but for picture I just show it like this. And then this can be attached to a single-stranded DNA, and the dye can be attached. And under our microscope, this is what we monitor on the computer screen. We can look at hundreds of molecules per movie, and their emission is quite uniform. When a protein is added to the system, and if the protein happens to interact with the DNA, then the emission of the dye becomes much higher. So even with the naked eye, you can already see that the molecules look brighter. You see, right? So about the intensity between this and this is typically about 2 to 3-fold different. So we get a lot of enhancement of the fluorescence when a protein comes in the vicinity. So this is hence the name protein-induced fluorescence enhancement. If you're wondering about how this works, so the dyes that work for this, what we call PIFE assay, is typically two like Psi dye, Psi 3 and Psi 5 is great for this. They have 2 rings interconnected with the carbon-carbon double bonds, and they isomerize. These two rings isomerize. And so what seems to be happening is when there's a viscosity object, such as a protein, approaches this organic dye, then it tends to bias the isomerization into the photoactive state, which is the trans state of the dye. So there's a photochemical basis for this. And so this is related to lifetime of the dye and also quantum yield of the dye. Yeah, so this, not in this paper, in another paper, we characterized the distance sensitivity of this assay. Okay, so that's the one-color assay. Now moving, so this is nice because for individual molecules, we can study the protein binding and dissociation kinetic at the level of single molecule, right? So we can combine all of these data and learn about the binding kinetics, on rate, off rate of the proteins, or even affinity of the proteins. So this is the paper where we characterized the distance sensitivity. Okay, so this is really handy. We call it a quick and dirty method. Typically a collaborator sends us a protein sample. Do you have a question?
Yes. This protein is said it enhances the fluorescence like 2 or 3 times. So what is the typical size of this protein compared to the fluorophore? And so if you did the same thing by using a better fluorophore, is that all that's being enhanced, the fluorescence?
You mean can you go higher than 2 to 3-fold? Is that what you're asking by changing the dye, or?
My question is basically, is that in this method, PIFE,
Mm-hmm.
we get 2 to 3 times
Yeah.
enhancement in the intensity of the fluorescence. So is that all that we gain? Because then if that's the case, wouldn't be, if there were a fluorophore out there that basically fluoresce 2 or 3 times more, if the result is the same. That's the first question. The second question is, what is the typical size of this protein compared to the size of the fluorophore?
Yeah, so proteins, we've it's not protein size dependent, so it's really dependent on how the protein interacts with the dye. So if the protein comes and binds in the vicinity of the dye, we get a really high fluorescence increase.
I want to see the invasiveness of this, basically, addition of this protein to this biological system. So that's one.
Invasiveness of the protein too.
It's a big protein.
Yeah.
And you're trying to study some specific effect. How does that perturb the system you're trying to study? That's why I'm interested in.
Yeah. So the question of interest is how the protein interacts with the substrate, right? So we're using this as a probe to report on that. So this assay really is so we're not really studying the photochemical effect. We're studying the protein DNA interaction, and it's really not size dependent. We've tried anywhere from 15 kilodaltons to 120 kilodaltons. These are typical range of proteins, from small proteins to big proteins, and we almost always get this about 2 to 2.5 fold intensity increases. And there's more to it than what I just show. It depends on the types of the interaction with the proteins, but we can talk about it later, or you can read our paper to learn more about it. But it's really handy because we get the enhancement when the protein binds. When it falls off, we recover fluorescence. So our data typically look like this. So before I go into this, imagine the protein is going back and forth, or it's binding or unbinding rapidly. These kind of things can be also made out because of the intensity changes that we can read out. So, and this is a 2-color assay, which I'll talk a lot more about in a few slides. So FRET stands for Forster Resonance Energy Transfer. So for this, you need 2 dyes. You need a donor dye and an acceptor dye, and you can position it at places that you desire. So in this case, we're depicting a protein labeled with a donor dye, green dye, and DNA labeled with a red dye. And in this case that we presented here, the protein actually did the acrobatic movement. It went back and forth on the single-stranded DNA multiples of times without falling off. So in that case, our fresh signal looked like this because the dye-to-dye distance changes periodically. So your protein goes and then moves back, goes back again, moves back. So this is the motion that we observed by FRET, and you can really generate a beautiful trace if the molecule behaves this way. So that's a demonstration of FRET. And this is a usage where the single molecule pull down assay was really handy. In this case, we were hoping to pull down telomerase, which is a huge complex enzyme from cell lysate. So typically we rely on this technique when the protein size is too big or it's hard to express and purify from bacterial cells or insect cells. Then we can overexpress this protein in mammalian cells. And the way we pull it down is we use antibody. So in this case, we had a flag, which is a peptide tag you can attach to your protein, and you can express your protein with this tag. Is this too much? You guys are okay. Yeah, so we can use an anti-flag antibody at the bottom, capture the protein, which is flag tagged. Then we confirm the presence of the protein with yet another antibody. So it's sort of like ELISA, if you are familiar with that assay, but done at the single molecule level. So in this case, it was successful. We were able to see a very good capture of telomerase from cell lysate. Okay, so that's a pull down assay. And then sometimes we go into cells, but again, our interest is to visualize individual molecules. In this case, we are visualizing, the image is kind of fuzzy here, but individual bright dots represents individual mRNA, messenger RNA inside of a cell. So what we do is called a single molecule fluorescence in situ hybridization, FISH. So basic idea is that individual mRNA gets decorated heavily by about 30 to 40 fluorophores. So these black line represents oligonucleotide or DNA strand that would be complementary to your mRNA target. And each individual one is fluorescently labeled. And you prepare 30 to 40 of these probes to bind to individual mRNA. Therefore, it's very bright in our imaging microscope. So this is useful for quantitative analysis of mRNA expression in cells. So this allows you to look at cell-to-cell heterogeneity. For example, this cell might be expressing a lot more than the neighboring cell, so you can make out the single cell gene expression this way. So we use this for looking at oncogenes and tumor suppressor gene expressions, or some immune responsive gene expressions. Okay, so I'm going to go a little bit more into FRET because that's a primary assay that I'll use in this talk. So how do I look at time? Is there? Oh, okay. Okay, so this is a distance sensitivity of FRET. So as I alluded to, we use a couple of dyes, one called donor, typically in green, the other called acceptor in red. So when their distance is close together, we have high resonance energy transfer, which is in this level. So this is the distance axis. This is the efficiency of FRET. And as their distance get farther and farther apart, you have lower and lower FRET, right? And so what we image is the intensity of the donor and the acceptor, and then by dividing the acceptor signal by the sum of the two, we get the FRET efficiency. And if you look at the distance sensitivity, it's sensitive in this 25 to 75 angstroms, or 2.5 to 7.5 nanometers. So that is the FRET sensitive detection range, which is ideal for probing biomolecular interactions. Okay, so here is where, so if you remember this Korean pop artist, Psy, became really, his YouTube video of Gangnam Style became really viral, right? You guys all watched it, right? Yeah. So a student of Takji Pa, who's in physics here on campus, made this Gangnam Style depiction of how FRET can be used if the dyes are attached to the singer's body. So here I think I have a video here, but I don't think this, oh, maybe the sound will work? Yes? No? Okay. Maybe it'll not work. Oh. All right, so you probably were all imagining, okay, that's a follow, we won't watch this part. So here's the FRET. So when the FRET dye-to-dye distance is about intermediate, you get a mid-FRET, followed by a low FRET when the dye distance is far apart, then a high FRET when they're both close together, then red dyes typically photobleach early. So when red dye photobleaches, the experiment ends. That's a photobleach state where you only have green dye. So if you were to draw a single molecule trace of this dance, it would have looked like this in our IDL MATLAB process data. We would be observing the intensities of green and the red in time sequence. So then we can depict, the motion. Deduce the motion based on the intensity changes. So I give middle school outreach, my students do, and they learn about FRET. And what we do is, it's the most popular module that we have. We bring glow sticks of different colors, red and green, and we turn off all the lights, and then they get to create their FRET dance, right? So it's a high FRET, low FRET. So they attach it in two different ankles, or sometimes neck, sometimes wrist or ankles, and then they do. So we give them traces to dance out, and so we turn off all the lights, and then they perform their FRET dance, and the rest of the audience have to guess the FRET trace, how the FRET would look on their sheet, and they get graded. They get scores, and they get snacks afterwards. So so it's really handy. We can do it on humans. We can do it on molecules. Very useful. All right, so what we actually do on the slide is this. We use quartz slide. We put down a silane layer, silanize the slide, and then we put down this polymer brush layer made up of polyethylene glycol. And this is crucial for rejecting non-specific binding of sticky proteins. So this takes a long time. but it's worth it when we are working with proteins. And then a small percentage, about 1% of PEG is biotinylated. Color is kind of faint. So that we can attach streptavidin layer on top. Then we can immobilize biotinylated molecules of your interest. This could be biotinylated DNA or protein or RNA or protein complexes. So we have this biotin, streptavidin, biotin sandwich layer. So here I'm depicting a DNA with a red dye. When this is ready, we can flow in protein of your interest that's supposed to interact with this DNA, which can be labeled with a donor dye, and then illuminate the surface with a laser beam. Then it creates what's called the evanescent field, and this thin section is where we take the intensities of donor and acceptor. And so you can see that all the protein that's floating outside of the evanescent field is not captured in our detection. So that's kind of an automatic way of rejecting the background fluorescence. So we only observe the ones that come in contact with the DNA. And this works really well. Okay, so this is a simplified view of our experimental setup. So we have, start from here, that course slide with the samples lying on top or at the bottom. There's an incoming laser beam, outgoing laser beam through the prism. And this is where the evanescent field is created. The lights are collected through the objectives, through the lenses and filters. The donor and the acceptor beam is split by dichroic beam splitter. And then they get recombined and projected into a single CCD camera, such that we can monitor donor and acceptor molecules side by side on our computer monitor. So what's in the circle is the co-localized, or they're the same molecules that emit donor and acceptor, and we can get the FRET efficiency by this simple formula. Okay, so now I'm going to give you a little bit of biological background. So RNA interference, how many people have not heard about RNA interference? You guys all? Okay, great. That's good. So RNA interference, of course, the Nobel Prize was awarded to the two founders, Andrew Fire and Craig Mello. It's, as the title, this is their seminal paper that led them to win the Nobel Prize. It's a way of potently and specifically interfere the genetic expression of RNA by treating the cells with double-stranded RNA. So what you do is you prepare double-stranded RNA that matches the sequence of your target mRNA, put it into your cell, and somehow magic happens, then your gene will be silenced, right? So they were the first, they're not the first ones to have observed it, but they discovered a pathway of RNA interference. Because it's potent and specific, it holds a lot of promise for not only for research, but for biomedicine as well. So there are many diseases. So if your disease comes from overexpression of certain genes, like oncogenes or cholesterol genes or any harmful genes that your disease is due to, then you can think about using this approach. So there are many diseases that are being targeted, including infection is one big area, cancer is another area, and other neurodegenerative diseases, where again, the overexpression of a certain particular gene is a problem. So many of them are under clinical trials. So why are we not using this more in clinic? That was actually my question when I first learned about this pathway. It turned out that it's not that easy. And one of the major known problem in RNA interference is this off-target effect, right? You design your RNA, but it does not go to your target. It goes elsewhere and targets other genes, right? So that's the off-target effect. Coinciding with it is also on-target deficiency. So again, you design your RNA very carefully, but you don't hit your target, right? Your target is not degraded or not erased or removed efficiently. So of course, it would be nice to target both problems, but this is really difficult. Where else is it hitting is really difficult. So we are going after this on-target deficiency problem. How do we improve the RNA design so that we hit the right target with high efficiency? So that is our goal of this project. So that's right? How do we improve the RNA design? So this is my lab posing for Gangnam Style. Yeah, my lab member doesn't like me because I make them do these weird things. So the person here, Heron Ko, she really spearheaded this project that I'll show you. And this is in collaboration with our Jennifer Doudna, who's at UC Berkeley, and graduate student Marianne Kidwell. Okay, so I'm going to go over the pathway, how this small piece of RNA silences a gene. And it'll look complicated because of the number of colors, but there are only 3 players. So it's probably a good idea to remember these names, 3 players. So, okay, there are 2 pathways that I have here, but they're very similar. So siRNA stands for small interfering RNA. So this is typically given exogenously. You can design and you can put it into your cell. And like I said, it has to be double-stranded. Oops. Okay, sorry. Okay, and then here comes the big 2 players. So dicer is scissor molecule. It's an endonuclease that cleave RNA. And so this dicer, together with this cofactor TRBP, are the ones that recognize such an entity. So if you know your cell biology, we don't have that many double-stranded RNA in our body, right? All of our RNA is typically single-stranded. Of course, they form secondary structures, but vast majority of RNA in cell is single-stranded. So when such a foreign molecule come into the cell, then these are the major players that recognize it and bind it and process it. Okay, so this molecule does not need any scissor activity. So what happens is that it needs to separate between the 2 strands. These are 2 complementary strands. It needs to select what's called the guide strand from the passenger strand. So name kind of indicates its function. Passenger strands need to be ejected out. Guide strand needs to be kept for silencing of your gene. So it does the strand selection, unwinding and strand selection, together with this 3rd player called Argonaute. And then it goes, Argonaute actually as a main player, goes to the target gene, finds it, anneal, make a pairing between this guide strand and mRNA, and then there's a degradation machinery that comes and chew up the RNA. So that's how the silencing occurs. So it's very simple, and to me, it's still magical how this happens. It turned out in early 2000, people discovered that it is actually a cellular pathway that make use of this silencing mechanism. And it's called a microRNA. So as I said, it's very similar. The only major difference is that these microRNAs are genome encoded. So our genes have a lot of regions that code for microRNA. So once the transcript is made, so this is made in just a regular way, it folds into the secondary structure, and then it undergoes some preliminary processing. At this stage, which is called a pre-microRNA, it gets extruded out into the cytoplasm, where the same thing happens. Now TRBP dicer come, and here you need the scissor activity, the cleavage activity. So dicer gets rid of this loop. Then it looks somewhat similar to this, right? Then the same thing happens, unwinding and strand selection. Then again, it goes to a target gene. In this case, people have reported that rather than chewing up the gene, it represses the gene expression. So it blocks it from being translated into protein. So the effect is the same. Whether you chew it up or repress the gene expression, you don't get viable protein produced from this gene. So in both cases, you are lowering or reducing the gene expression. Okay, so our assay, I don't know how far I'm going to get to. I'm just going to stop when my time comes to stop. So we devised stages, single molecule and pull down and single cell stages to actually quantify each step of the way. And again, our idea is that by designing the RNA substrate with some better logic, perhaps we can have better efficiency at each step of the way that leads to efficient silencing. Any questions about this pathway? No. So I'm just going to show you the stages of testing, which involves a lot of the single molecule assays that I already told you about. So there's, again, our 4 stages of testing, and our main hypothesis being that RNA, it's already known that RNA structure plays a role in this, but we don't understand how exactly its length and secondary structures and having a loop versus tail contributes to the efficiency of RNA interference. So that's our variable. And these are the platforms that we will test the efficiency of silencing. Okay, so the first stage is to look at the binding, the initial contact between this protein and the substrate. I'm gonna probably skip through this because I'm gonna show you all of that. Okay, so the first thing we checked out was this partner protein called TRBP, if you remember. That was the second player that pair up with the scissor molecule. And it's known to increase the binding affinity of this scissor molecule to the RNA. So our simple idea was let's look at this binding event. How does TRBP contact double-stranded RNA? Does it do anything? So in this case, we labeled the protein with the red dye and labeled the RNA, double-stranded RNA, with the green dye. So our expectation was that we'll see FRET. Yes, as simple as that. And so if you look into TRBP, it has 3 double-stranded RNA binding domains. So it looks like it's destined to bind to double-stranded RNA. So when we do that, we saw a very unexpected result. We saw these huge FRET fluctuations, if you will. So this is the intensity of donor and acceptor, which is anti-correlated, right, because the distance keep on changing. And this is the calculated FRET. And notice that FRET does not go to 0. It's staying between about 0.3 and 0.8, which means that protein is not leaving the site. It's maintaining contact, and it's moving across the double-stranded RNA axis. So the protein binds, and then it goes back and forth in this axis. And we did a lot more work to show that it's actually the protein moving, not detaching and attaching. And we moved the dye distance around to show that it's actually the entire protein moving across the RNA. So this was very unexpected, and so the same thing can be shown in this one-color assay. So as you can imagine, if this is the RNA substrate alone that has a very stable emission, when you add the protein, then the intensity fluctuates. If you look at the level, it's about 2.5-fold. It goes from about 200 to about 500, right? So that's the fluctuation range. Yeah. Yeah, so our exposure time used for this assay is about 50 to 100 milliseconds. So we're not detecting anything at a microsecond level or anything like that. So that's about the level. And this movement is about 800 milliseconds or so, one cycle of going back and forth. So it's measurable.
On the previous slide, you showed that the red X-ray how do you know that that is true, sort of the true effect and not maybe under sampling or, because I could imagine if this is happening faster and you're sampling it within like 100 milliseconds,
yeah,
then this signal would be sort of undersampled, that it would look different.
Yeah, we always undersample due to the time resolution. Yeah, so as long as we can tell. So could it be possible that it detaches in, short, while it attaches back and goes back and forth? Yeah, that's possible. I would argue yes. Yeah. Yeah. Yeah, so the same thing can be shown in this one-color assay. We can even go into 3-color assay. We had to do this to satisfy the reviewers' complaints. So in this case, we have 3 colors. So we have 2 acceptors at both ends of the RNA, right? This is acceptor 1, this is acceptor 2, and the protein is labeled with the donor, right? So this is really to show that protein is indeed moving from one end to the other end. So 3-color FRET is very messy, as I'll show you. It's almost impossible to interpret. So, but what we are looking for is when there's this FRET, this is dark. When there's this FRET, this is dark. So we're really looking for the anti-correlated change between the 2 acceptors, right? So this is what we actually see. Signal is messier, but we do see that the two acceptor signals are anti-correlated, which tells us that the protein is indeed moving from one end to the other end and back and forth. Okay, so that's a usage of a 3-color FRET. And it turned out that, so then next thing we did was to vary the length of the substrate from short to long, and basically 12 base pair is way too short for the protein to bind, so we don't see any binding. 15 base pair is just enough for the protein to bind and remain bound for some seconds. Beyond 20 and 40, we see a stable binding and this sliding activity. And you can notice that the amplitude change is bigger in 40 base pairs because there's more room to move around, right? And then we tried out some mismatched substrate. If you recall the previous slide I showed you, a lot of the microRNAs actually have some mismatches along the stem. So it's not a perfect duplex, but it has some base pair mismatches, or sometimes some kind of little loops exist in microRNA. So we thought to vary those structures. And so minor disruptions or minor mismatches can be tolerated. Its protein still binds and slides. But if your disruption becomes too big, like the bulge or loop becomes too large, then the binding and sliding does not occur. So based on these examinations, we kind of hypothesized that this TRBP can play a scanning role. So it can, before it elicits the huge RNAi pathway, it can perhaps bind and check out the substrate to see if this is a correct one to be used for silencing. So that is our kind of bold hypothesis, that TRBP sliding may be sensing or scanning the RNA prior to activating the pathway. And it turned out that, so these are two other orthologs or homologs of this TRBP protein. They have a very similar molecular arrangement. They also show sliding. And since this time, we've seen how many more? About 4 more proteins of this family, which also slide. So at first, we thought this was a very, very unique activity of this protein alone, but it turned out to be a more conserved universal activity. And so we are studying more of these molecule families. Yes. Okay, and then we thought to introduce disruptions along the way, or barriers along the way, so to see how much it can tolerate. So we created some mismatches of different sizes and a bulge, which is just longer strand on this side. And we inserted DNA because I didn't get to show you, but this TRBP binding and sliding is very highly selective to double-stranded RNA. So it does not even bind to anything other than double-stranded RNA. So we thought to create some DNA strands into the strand. So how does the barrier effect look? This is the barrier, which mismatch turned out that it doesn't do much things, so it looks just like wild type. But the barrier 4, which is the bulge 4 of the bulge strand, induces some kind of stalling in the molecular motion. And DNA by far has the biggest effect. It kind of makes the molecule stutter in between on its path. So to quantify this result, we measure the sliding time in these different substrates, and you can already see that DNA plays a big role in interrupting this motion. Okay, hopefully you guys will stop me on time. I'm going to skip over this data. This is more about, and then we looked at the molecule, and it was interesting to see these two domains were connected by this linker, and it's a very long linker that we don't often find in these subdomain regions. So we thought we hypothesized that this may play a role in sliding. And so what we did was wild type has 61 amino acid, and it gives us this kind of full sliding activity. Then we shortened it to 37 and to 17. And it turned out that the sliding distance also shortens as the linker length shortens. So this tells us that perhaps this linker plays a role in inducing, or it controls the distance to which it can slide back and forth. So yeah, that was just one interesting spring-like amino acid that was built into the molecule. And then I'll briefly just show you this result before time runs out. So the next actually stage is to look at the dicing or the scissor activity, the cleaving activity. And for this, we need the scissor molecule, dicer. And so we prepared a dicer which has a tag and a TRBP that's hooked up to it. And this assay basically showed us that, okay, so what it is protein, in this case, protein complex was labeled with a red dye, and RNA substrate was labeled with a green dye. And we found about half of the molecule giving us the static fret signal, which means that there's no movement, and the other half giving us this dynamic motion, which meant that this protein as a complex is sliding along the RNA. Well, in this configuration, RNA is sliding along the protein complex. So then to induce the dicing or cleavage, we add magnesium, and it starts cleaving the molecules. And we counted the molecules of static versus dynamic, which one gets cleaved faster or slower. And it turned out that only so this is complicated to look at, only the dynamic molecules were selectively cleaved, right? So this told us that this sliding actually is facilitating the dicing reaction or the cleavage reaction. And this is kind of the novel part of the study that we came up with. Based on this model, we came up with this idea that dicer by itself, without the cofactor, can only bind. Dicer itself cannot slide. So when it's lucky enough to bind at the cleavage site, it can cut. However, if it binds anywhere else, its binding would be not productive. So it has to dissociate. Whereas dicer TRBP complex is able to slide along the RNA to search for the cleavage site. And then when it reaches it, then it can cleave. So we think this TRBP-induced sliding motion is actually playing a functional role, that it facilitates dicer to find the cleavage site more efficiently. So this could be a rate-limiting step. So we are, again, measuring the RNA silencing efficiency at different stages of the pathway, and this is the second stage. So our idea is, which RNA induces sliding or doesn't induce sliding. That would be the first stage of testing. Second stage would be which RNA gets cleaved faster or slower, because that could be a rate-limiting step in the pathway. So this is 2 different RNAs that we tested using this assay. So the difference is that this is pretty much duplex, no secondary structures. This RNA has a lot of secondary structures. It has some mismatches. It has splayed arm. Turned out that this is a dicing kinetic. Turned out that this RNA gets cleaved much faster than the other RNA, which is interesting. And it matches the biochemical data, so this seems to be the case. So it's not very simple, right? You would think that very non-structured RNA may be cleaved or processed faster, but it turned out that structure actually helps. It speeds up the processing at this stage. So we're examining many different substrates to measure these kind of dicing kinetic. Yeah. Did you have a question or no? Okay, I think, yeah, so I have 2 more stages, but this is too long, so I think I'll just stop here, and if you have questions, I can take more. Yeah. 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 Mm-hmm. Well, if they move, you mean the fluorophore and the protein moving together, then the intensity shouldn't change, right? It depends on the distance between the protein and the fluorophore in that case, right? Yeah. Yeah, then we can't tell. That's why we don't. Right, so it can change binding position without changing the intensity, right? You're depicting that situation. That's true. So we don't really rely on one single assay for our assessment. We typically use both FRET and PIFE, because sometimes there was a very nice case where we were looking at protein binding, and if you imagine DNA going this way, this protein binds and forms filament, right? So this protein nucleates somewhere and then it forms filament in one direction. So when you image it by FRET, so we attach two dyes like this, and then when the DNA is by itself, it's flexible, so it's high FRET. When the filament forms, then FRET decreases because the dye-to-dye distance keep on changing. So we have this FRET step down, but that assay didn't tell us which way the protein was forming. Is it from this way to that way, or is it from tail to this way, right? By using PIFE, then we attached one dye here, then we saw this stepwise increase, right, of the intensity. When the dye was here, we saw this one-step increase that remained the same because the protein filament grows from this direction to that direction. So that's a classic case where we use two assays to get a more complete idea. So sometimes it serves as an alternative FRET, because sometimes protein is difficult to label. It's very labor intensive and it's very frustrating. But this assay could be a quick tool to use. Yeah. 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 So PIFE, if there is can I use, sorry. Maybe this, can I use this? Yeah, I'll just be quick. So FRET, it's about 2 to 8 nanometer, and it looks like this, right? FRET sensitivity. PIFE looks like this. So you actually gain sensitivity in this very small distance range, like 1 to about 3, 4 nanometers. So this is another benefit of using both assays to get a complete idea about this whole range of distance. So we use it for that reason too. So that's a good question. That's already published, so you can look up more. Yeah.
So the way that you show the dynamics change when you do secondary structure.
Yeah.
The sliding movement is effectively a way to read secondary structure. So my question is, if there are no variations of TRBP or its analogs to read like microRNAs differently, especially in the cold blood dynamics.
Yeah, that's a really great idea. So we try to also look into our traces to see if there are any differences, but yeah, we couldn't spot any. The time-wise, the periodicity didn't change much. So we went to the lower temperature to slow it down to see differences better. But that's a great idea. We think there could be some differences that we're not sensing. So yeah, and we're examining a whole bunch of TRBP-like proteins to cross-compare and which ones slide better, which ones less, and all of that. So yeah, great question. Yeah.
So kind of going back to your instruments, is there a reason why you use more as your?
Oh, that's, yeah. Anyone else? So it's for the refractive index. That's the only reason. Yeah, for TURF, total internal reflection measurements. Yeah.
Is there a reason why? Do you have any sort of between your regulated biochemical DNA and
So the polymer is pretty mixed length. So yeah, we could use a uniform length, but it didn't really matter. So it's, yeah. So there's not a set distance. In fact, we did some AFM imaging to look at the surface. It's pretty rugged and messy. Depends on who makes the slide too, but it's not that homogeneous. So that impacts signal strength sometimes. We see some differences in signals in neighboring molecules, and it can also be that the height is different and the where it sits at the evanescent field is slightly different. But in most cases, our intensity histogram is pretty sharp, so we think it's okay. Oh yeah. So are you talking about molecule interaction between DNA and the PEG? Or no?
No, just like so since you're red, does this between each of the protein binding with our DNA is the difference between the distance DNA and PEG?
Oh, yeah. That's an important question. So we seed our DNA at a very low concentration, typically picomolar range. So this is 10 to the minus, 12. So that's very crucial. So we cannot go very high. So we seed it so that they will be well separated from each other, at least. But, yeah. So you've seen the image, and that image, the width of that image is about 25 microns. And you don't, across the 25 microns, you don't see very many molecules. I would say maximum of 20. So yeah, it's that density is crucial. Thank you for the question. Yeah.
So on your, when I was doing like your frag and your pipe over time, the intensity profile, you try to do like sort of like spectral analysis, PCA or something to see if you get that, you can go backward and find out what the problem is.
Yeah. We've done some of that. Mostly proof of concept to see if this method is good for doing this, for like the filament forming proteins.
Yeah, but like you were able to go backward and say, oh, this one has 2 mismatches, this one has.
Oh, mismatches, yeah, not to that degree, unfortunately. Yeah. But we are trying. These are very, great questions. You guys are getting to the core of our research. It's great. The other, yeah. Oh, so we have typically about 15-17% leakage, donor leakage into the acceptor channel. So yeah, there's that. We can separate it more, but then for the FRET imaging, you need spectral overlap between the donor emission and the acceptor absorption. So yeah, you have to find that sweet spot where you minimize the leakage and maximize the FRET efficiency. So side 3 and side 5 in most cases that we use are kind of ideal to meet this criteria. But we're looking for better dyes. Alexa dyes also are good. Atto dyes are used. Yeah, these are good FRET pairs. Yeah. It's thought to be oxidative damage, right, on the molecule. So yeah, side 5 is, if I talked to you about the 2 rings interconnected, it's much longer interconnection. So it's in isomerization, maybe it has a more chance of just being going into the dark state or these states. You guys have a question? Or? Oh.
What's the typical temperature and pH when you use this?
Yeah, so.
How sensitive is it as a method to each of those?
So we typically use room temperature. So all of these assays were done at room temperature. So some of the cleavage assays, we go up to 37. So it's typically between 22 to 37 degrees. For some proteins, we have to go up to the physiological temperature, body temperature, 37. Up to that level, fluorescence intensities become slightly brighter at higher temperature. But in FRET imaging, they both become brighter, so FRET efficiency is not changed. pH 7 to 8. We can go down to about 6. We don't like going down to 5 because things don't function. Yeah. Yeah.
Have you ever made proteins that would still have efficiency in lower or higher pHs?
We did, and I have a faint memory, but it was a protein that we had to go down pH 5. That was troublesome. So that's why 5, we try to stay above 6. 6 to 8 is fine. 8.5 is fine. Yeah. That's a good, very practical question. Yeah. Yeah. How many molecules do we get for? Yeah, typically, high throughput, about 300 to 400 molecules. Yeah, it depends on the density. So I know some students typically have like 600 to 700. They're reaching the limit of not being able to detect single molecules. But healthy range would be 300 to 400. But protein binding events may be more rare depending on the protein concentration, that type of thing. But it's a pretty I mean, compared to like optical trap kind of assays, it's a truly single molecule assays. This is much higher throughput. So yeah, we typically look at the intensity profile first of the donor and acceptor and Fred, and then visit the individual molecules. We also carry out ensemble measurements always on the side, so to cross compare, because in case single molecule introduces any artifact, we do that. Yes, we want to use it. Yeah. We just haven't made the progress into that direction. But it's, of course, would be ideal. Yeah. Any other questions? Dr. Kongmer might be. All right. Yeah, very, very nice questions. Thanks.